Right, not that long to go before paper 2. Let's bust out biodiversity. Biodiversity, module 4 topic. Okay, it does have a lot of connections to populations and sustainability. So I think that's the reason why these two are on the paper. Perhaps there's going to be a question with overlapping those two topics. Okay, so biodiversity, we begin with the levels of biodiversity, right? So the types. So what are the types of biodiversity? So that's the first little bit. And we kind of keep coming back to that idea. So once we've established the types, then we really get into measuring. Right? So measuring, understanding how we carry out sampling, right? So measuring biodiversity, that's the other main topic in this area. And once we've done the measuring, we generate some data and we have to analyze what that means. Okay? So we have the different types of biodiversity we're going to be measuring. In fact, we could just get into it, but let me just kind of broadly speaking, put everything out properly so that we have enough space to do everything right. Okay, so in measuring, we are going to be measuring species biodiversity, okay, and we'll also be measuring genetic. Diversity. So that is in fact a bit of a spoiler for the types of biodiversity, so the levels at which biodiversity exists. The three types are habitat, species, and you know, do try and put that, it's not just three types, they are, so you know, the whole planet has different habitats, each habitat has many species, and many species are individually populations, and within each population there exists genetic diversity. Okay, so we have habitat, species, and genetic. Okay, and again, that is, it's not just there. That idea is all the way through this module or this topic. All right, so we've got these three types, and now let's go into measuring species diversity. All right, so in terms of measuring species diversity, we are talking about sampling. Okay, now remember, the core idea of sampling is you can't go out and count every single organism that there is in a particular area. So what you do is you take you sample a representative area and make some extrapolations in relation to the bigger area that you're concerned about or concerned with. Okay, so in terms of sampling, we're going to first think about general ideas about sampling, okay? Because in sampling there are two main things. There's sampling for plants and there's sampling for animals. All right, and your approach or your methodologies to do these different types of sampling is slightly different, but the underlying or the overarching principles of how you investigate or do sampling in plants or animals, those are the same. And what we'll do is just summarize that as strategies. Okay, so strategies these are the different types of approaches that you can take to sampling. What you must always try and have is to have something that is unbiased. Okay, so whether you want to do it randomly or systematically or stratified, that's kind of a different way. But those are all ways that you can be unbiased. So unbiased doesn't always mean random. Okay, though random is one way that you can introduce or avoid bias. Okay, well, the key principles here are we need to be unbiased, and we need to have a sample an area that is in some way representative. Okay, now so these are the critical things that you have to have as part of your your strategy, your sampling strategy. But there are approaches that you can take. So what are those approaches? So those approaches can be random, right, which we'll see in a bit later. But usually random means that you're using your random number generator on your calculator, you're generating coordinates using that, and you're using those coordinates to direct you towards which areas to concentrate your sampling. Okay, so I will random, and I'll just put here random numbers, and numbers should relate to some kind of coords. That's short for coordinates. Right, the other way you could do it is the non-random approaches. Okay, yeah, so we could have random, or we could have, I'll just put it down here, we could have non-random. Yeah, now non-random means, or under the umbrella of non-random, there are different ways to be non-random and unbiased, okay, and that generally means you're kind of grouping things and organizing things, so doing things in a systematic way, right? So every so much distance I will take a sample, right? That's not random, but it is also unbiased, okay? So you could be systematic in your approach, as we take usually with transects. We could be stratified. Okay, so in order to sample things in a representative way, you may want to divide certain things into categories and make sure you take a sample from each of those categories, okay, to avoid missing out any large number of areas, organisms, etc. Okay, so we can have a systematic approach, we can have a stratified approach. What is the other one? Systematic, stratified, and opportunistic. We're saying that systematic is okay, stratified is okay, optimistic, it's not optimistic, opportunistic is something that we're kind of not so sure about, that introduces some form of bias. We can then move on to the methods to sample these organisms. So in terms of plants, we usually, the approach that we take is either an area comparison. It's an area comparison, i.e. this area A versus area B. So that could be 10 meters. So that could be 10 by 10 meters, and that could be 10 by 10 meters, and one could be in a shady area, one could be in a well, in an open area, one could be in an area near the road, another area can be quite a distance away from the road. So what we're looking at is just two areas, one versus the other, and in that case what we would use is quadrats. Okay, right, so we could have an area comparison, but we could also have transects. We can also have transects. So transects is usually whenever you want to measure the biodiversity over a linear distance, okay, over 10 meters, 20 meters, 100 meters, but you're interested in how things change along a particular distance. So generally, transects is when you are thinking that there is some kind of correlation between an abiotic factor and biodiversity, the number of a particular species, the abundance of a particular species, etc. So transects are when you want to see whether the abundance of a particular species is correlated to an abiotic factor. Okay, yeah. So with plants, what we usually do, so the area comparison and the transects can be used with quadrats. Okay, so usually if you're looking at very short, like mosses, grass, daisies, that kind of thing, a quadrat is fine, like a frame quadrat. Okay. Right, but there are different types of quadrats. If you're looking at very long, like tall grasses and tall plants, things like that, not trees, right? There are modified versions of quadrats called point quadrats, right? Which are these frames with needles hanging down, and whichever plant is touching the needle, you count that and use that to come to the abundance measurement. Okay, so we got quadrats. Transects can be used with quadrats, and in the case that a transect and a quadrat are used together, that is called a belt transect method, and it's usually used with a systematic approach. So it's not random along the line, it is every 5 meters or every 3 meters, every 10 meters, etc. Okay, remember that, if you're sampling over 100 meters and you're only taking 5 quadrat measurements, that might not be representative enough. Okay, so have an idea as to what would be representative, but it should be systematic. Okay, with transects, we could also have line transects. So that's simply like a tape measure in a linear distance, and we're not using quadrats, but at certain points along the tape measure, we are seeing what is touching the tape measure, and that would be a line transect. We'll come back to what we record and stuff like that. Let's now quickly look at animals. So what we're just looking at right now is what are the tools? What are the kind of different approaches the different tools we might use to sample plants, and the different tools we might use to sample animals. With animals, we're looking at like small creatures, though it is just like OCR to teach you about sampling woodlice and then ask you a question about panthers. Yeah, that's nice, isn't it? That's very compassionate. So animals, we have small, so it's usually like small animals. So what are the things? So I don't want to go into too much detail here, but I'll just point out some of the examples of things that I would like you to think about. Remember things like pooters. Yeah, that's a thing. Pooters, pitfall traps. Yeah, pitfall traps, pooters, kick sampling in riverbeds. Yeah, what am I missing? Tree beating, right? Okay, but these are all the ways that kind of tools that you could use to measure small animal life. Yeah, woodlice, moths, ants, beetles, that kind of thing. But I guess the idea would be that if you understand the principle behind that, I guess you could take the core idea of that principle and apply it to organisms of any size in any location, theoretically. Okay. Right. So so we looked at how we look at how we analyze how we sample plants and how we sample animals. But in general, what is it that we have to do then? Okay, so we've got our strategy, we've got our tools now. What do we do with that? We record. So what are we recording? And that is a key feature here. So what you're recording is really, what you're interested in is richness and evenness. All right, so richness and evenness, when you put those two together, when you sample, when you record, which organisms you saw, how much of those organisms you saw, that essentially equates to evenness and richness. And when you do that, then you can put those numbers together and you can generate the Simpsons index of biodiversity. And that's really what we're working towards here. Okay, so in terms of richness, what you need to do is identify species. Identify the species present. Okay? When you're looking at evenness, what you're really interested in is how much of each species is present. Okay, so that is you estimating or counting abundance. So you can either count directly if your organism is easily countable. That's the ideal scenario because you get exact numbers. But you could estimate. Okay, so especially for plants, you could estimate, you could do percentage cover. Right? So if each square of a quadrat is a certain percentage, you simply count the number of squares in which that species was present and it gives you an estimate of percentage cover. Remember that you do want to ultimately measure this per. You want to average. So you've got a number of different quadrats, obviously, in a particular area. Overall, you want to get an average of all the quadrats per meter squared. All right, so your abundance, you want to get to there. All right. Richness, you want to identify the species. That's fine, good. Now, in terms of evenness, so we can identify animals based in a number of different ways. But in terms of animals, in animal abundance, remember we have the mark and recapture technique. Mark and recapture, and that allows us to measure animal abundance. Okay, and I would like you to apply this technique, however ridiculous it seems. But remember that OCR has given you some basics. Your teachers have provided you with some essential concepts. You don't have to go into the biology exam and create something completely brand new. Okay, you're not sitting an exam for a Nobel Prize. You're going to take these ideas and you're going to apply them the best you can, even if it sometimes might seem a little bit silly. Okay, so mark and recapture, there is an equation, so there's a technique, so there's a kind of a series of steps that you need to do to be able to generate the data for this. So, capture certain, do one round of sampling, trap certain organisms, mark them, release them, allow some time to pass, set the traps again, recapture, see how many have been already marked. So there is that, and there is an equation that you can use to use the data that you generated from that to give you an estimate of population numbers. Mark, release, recapture, mark, release, recapture, equation, estimate total population. Okay, and that, once we've got evenness and richness, that then allows us to deal with or generate a Simpsons index of species diversity. All right, and all I would say in terms of Simpsons index is, yes, if you're not too sure about that, you've got time, you can practice a question or so. There is the equation here. Now you should be given this equation in the exam, I believe. I'm not even going to bother writing it. I'm not going to bother with that. Okay, so Simpsons index, know the maths, know how to do it, but what comes up quite a lot is interpreting the value. Okay, so remember, 1 should be the high biodiversity, and it goes all the way to 0, indicating a low biodiversity. Yeah, okay, but whatever numbers you get, push comes to shove, you get a number that's not within 1 and 0, just maybe that means something's gone wrong in your calculation, so check it. But if you're really pushed for time and you don't have that time, just remember high value, high biodiversity, low value, low biodiversity. Okay. And I guess, I mean, if the biodiversity value is low, that does then take us to conservation, which we'll be looking at over here, right? So low values, low biodiversity means conservation necessary, because what we're really talking about is ecosystems having low biodiversity. That means the ecosystem might be under threat, and loss of an ecosystem is very disastrous. Okay, all right, because we rely on ecosystems for a lot of things. I did miss something out, and I did want to say something here also. Right, plants, correlation. Now, usually, what you're trying when you're sampling plants, you're usually interested in how some kind of factor. Now, the area comparison, it might be that, or it might be the correlation. You're usually investigating the effect of an abiotic factor on plants. So I might just put that here. Whether it's quadrats, whether it's area comparison, or whether it's correlation, you're usually trying to analyze the effect of some kind of abiotic factor. So be aware that you could be looking at pH, you could be looking at water availability, it could be humidity in the area, light intensity, those things that you would expect to affect plant life. Okay, so there is that. The analysis of the data, it might not just stop with the Simpsons index, however, because if you're comparing two areas, remember, that could involve area comparison, could involve doing or analyzing your data with a t-test. All right, and if you're doing a correlation between an abiotic factor and the species abundance or biodiversity, you might then have to test that correlation statistically with a Spearman rank correlation coefficient test. So do look over, I don't care how much time is left before the exam or when you're watching this, but do give that a once over. It might make a difference, right? Grade boundaries could be razor thin. So in that case, even though you're thinking, well, what's the point now? There is a point, because a few marks here and there could make a lot of difference. Okay, so t-test, Spearman rank correlation coefficient, but it is module 6, so make sure that you also have looked at chi-squared test. All right, let's quickly move on. Genetic diversity. Yeah, now genetic diversity is all about the number of the different alleles in a population, the different alleles in a population. So more heterozygosity means more genetic diversity. Okay, so watch out for this. This can be presented to you in a load of different ways. They're not going to come out straightforward and say heterozygosity equals genetic diversity, calculate. Okay, so they might present to you anything that looks like, you know, what you're looking for is this, for the same gene, are they giving you information? Are they giving you information about different versions? This could be genetic profiles, right? Where for a particular locus, right? One locus, or for a particular gene or for a particular locus, you've got version 1, version 2. Version 1, version 2, i.e., it might be a genetic profile, right? Where some people have 1 band and other people have 2 bands. Well, the people with 2 bands are heterozygous. The people with 1 band are homozygous. Okay, it could be. Just look out for any version of that. All right, and yeah. Okay, so we're looking for the index of heterozygosity, or I think in the OCR textbooks, it's referred to as the proportion of polymorphic gene loci. All right, so a proportion of polymorphic genes, that's what it was, divided by the total genes. That's what it was. All right, and that should give us an idea of genetic diversity, and that's how we measure that. Yeah. Okay. Though, yeah, because it's module 6, we could get something to do with electrophoresis. I mean, would I be surprised? I would be surprised if you had an in-depth question on electrophoresis, though we can't rule that out, but maybe the electrophoresis shows some kind of genetic profile, and from the genetic profile we might have to determine which are heterozygotes and so on, and determine the index of heterozygosity. There have been questions like that in the past, so maybe it's worth looking them up. Okay, right. Now, it is in the textbook. It's not specifically on this part of the specification, but there is something to do with factors affecting genetic diversity. So I'm just going to put that here. I'm not going to list all the factors out because there's quite a few, but there are direct connections to other topics in module 6 which aren't on the advanced information list, but we have seen that, you know, there are things coming up that aren't technically on the list, though they have been small in proportion. Okay, but I think it's worth looking at. All right, so there's factors affecting genetic diversity, things like genetic drift, things like the founder effect, things like selective breeding, things like that. Okay, so the factors that affect genetic diversity are worth having a look at, even if it's just key points and you're just aware of them. Okay, so there is that. So next we're going to go to factors. We're going to go back to species diversity, and we're just going to have a quick look at the factors affecting that. All right, so be aware of the factors that affect species diversity. There's 3 main ones: deforestation, agriculture, hopefully I've spelt that right today, deforestation, agriculture, and climate change. Okay, interestingly, all as a result of human population growth. So that's an interesting connection to population and sustainability, right? Yeah, so factors that can affect all due to human. So these are human population impact. It's not just that humans are making poor decisions. It's not just it. It's about the human population is growing, so we need resources, so we need food, and as a result of our energy needs, potentially the climate is impacted because of that. So there are these things. What I would have in your mind is some clear points about how deforestation impacts on genetic diversity, on species diversity, and the implications of that. Okay? Agriculture. So not just be aware that these are the factors, have some points about what that means specifically, and some explanation about how deforestation impacts. So have that connection in your mind. How does deforestation reduce genetic diversity? How does agriculture reduce species diversity? How does agriculture reduce genetic diversity? Have those arguments in place. Right, then we move on to maintaining biodiversity. Okay, maintaining biodiversity, and the way we do that is conservation. Right, but so the maintaining biodiversity, there are reasons. Okay, yeah. So the reasons for maintaining biodiversity, right? Aesthetic, ecological, economic. Okay, I'll just give examples, right? So for example, in terms of ecological, our ecosystems, the more diversity they have, the more they can adapt and almost absorb the changing environments and things like that. But you reduce the species diversity, if you reduce genetic diversity, if the populations become so small that the genetic diversity reduces, we don't have enough variation to be able to then possibly cope with the changing environments. If we've got large amounts of genetic diversity, it increases the chance that there's individuals that can cope, that can survive with the changing conditions, allowing the populations to continue, or the species to continue. Okay? Economic, remember, we rely on our ecosystems for a lot of natural resources. Okay, so this is not just an ethical argument here. Our lifestyles depend ultimately on natural resources, food, building materials, etc. And finally, aesthetic, and that's mainly to do with, well, that's kind of connected to the economy through tourism, but it also has impacts on health. Yeah. Okay, so then we go on to conservation and the methods of conservation. And conservation in situ, right? In situ, and we're talking about in situ animal conservation. So what are the examples? We're talking about in situ plants and ex situ examples of what is the conservation ex situ for animals? What is the conservation ex situ for plants? Right? Now, in this case, you need to know what those examples would be, but I would also say, for each of these things, know the advantages and disadvantages. So for all of these things, ex situ animals, in situ plants, all these different combinations, whether it's wildlife reserves, whether it's botanic gardens, all of those things, know how they operate, but also the advantages and disadvantages in all cases. And finally, in terms of conservation, we do have the agreements. Okay, again, right now I'm just going to point these out. just have an idea of A, their primary objective, and B, just have an idea of A, what they are, and what their individual objectives are, because they are different. And yeah, they might seem to be working towards generally the same thing, and that's true, but they're each doing it in very specifically different ways, and that has come up in questions in the past. Okay, so we have the main things are sites, we have the RBD, the CBD is the Convention on Biodiversity. I always call it the RBD. Okay, we have the CBD, Convention on Biodiversity, that's about sustainability. Sites, trade in endangered animal products. And finally, we have the countrywide stewardship scheme, so that's more UK based. Yeah, or is it England? Quick note on sampling, keep coming back to this. Where we put the quadrat down, yes, we record which species is present. Yes, we record how much of that species is present, absolutely. But remember, we're also, in case we're investigating an abiotic factor, we do need to be recording the abiotic factors as well. So on our little results sheet, for each quadrat, we're going to have species present, number of or abundance of the species, and soil pH if we have a soil pH sensor. Or we're also going to be recording, all right, what's the light intensity in this area? Okay, because whatever our hypothesis is, if we're hypothesizing that as light intensity increases, the biodiversity increases, we have to measure light intensity to be able to confirm that, but we also have to measure the other factors just to confirm that it's not any of the other factors that are impacting on the, or also correlated with the biodiversity or the species abundance, whatever it is. Okay, so just a quick note on that sampling involves species evenness, species richness, yes, but also the recording of other abiotic factors where possible. So there are things such as light meters, pH sensors, soil moisture sensors, which thermometers to measure the temperature if that's what we're investigating, humidity level or measures or meters, right? So there are those devices available, and you can use those or incorporate them into some hypothetical method if you ever needed it. And I think that's all I really want to say about biodiversity. Trying to do it quickly. I know that the exam is not too far away, so hopefully that's kind of been reassuring that you've covered all that stuff, or B, hopefully it's helped identify some areas that you might need to still look at. Okay, right, good luck.